Set up a passive outside Probably not. I'm not really sure what to tell you either.
Our next presentation will be modeling regional distribution using acoustic telemetry, presented by Matt Futia from the University of Vermont.
Yeah, thank you so much everyone for coming in today. I feel like I need to start with an apology because I'm actually not talking about lake trout. So if you're hoping to hear some more about the exciting stuff going on with the lake trout population in Lake Champlain, there's a lot of folks here that are happy to talk about it, and if by some miracle I'll have some extra time at the end of the talk. I did throw some slides in after questions that we can get into that if there's interest. But yeah, so today I'm going to be talking about this modeling comparison that I've been doing, looking at regional distribution of fish using acoustic telemetry. I've been working with Tom Binder, Mark Henderson, and my advisor, Ella Marsden, and yeah, let's get into it. So acoustic telemetry is this great technique for studying the movement of fishes. You have a transmitter that you insert into the body cavity of fish, and that transmitter sends out a unique signal once every couple minutes or so, and that will get picked up by stationary receivers. And so you have this timestamp associated with a general location that a fish occurred at. And this allows researchers to study fish distribution from local scales to really these system-wide scales. So you can see that in the figure here on the right. These are all points representing acoustic telemetry projects, and this is already a little bit outdated, but you can see that this huge extent of projects going on. And with the collaborative nature of telemetry, there's a lot of resource sharing, a lot of data sharing, so that's where you really get into these system-wide movements that we can look at. And when you can really see the growth of acoustic telemetry in the Great Lakes here, so this is 2010 and the initial studies, and over time the studies have just exploded. And each circle represents a receiver station, and there's just so much potential now with all these different receivers out there, and this huge just system that's now available for researchers to become a part of. And in addition to this, there's also a lot of collaborative networks that have developed over time. So we've got GLaDOS in here in the middle, but globally there's a lot of other networks that are really enabling these large-scale collaboration efforts and a lot of data sharing and also resource sharing to allow projects like this to be possible. And this larger spatial coverage provides a lot of new perspectives to ask really important questions. So things like the invasion ecology and range expansion of different species, maybe biological and environmental drivers of animal movement, from a more applied or management perspective, cross-jurisdictional movements. How are these fish how are they moving around these different systems? And then also things like the distribution around areas of interest, and general range of target species. So I like to think about the American eel here and just such a large migration for these species that acoustic telemetry can really start to get into once you have these large collaborative networks and this huge coverage of receivers. But a lot of telemetry is dependent on, one, where you have receivers. You can see there's a lot of different types of arrays that are present just in the Great Lakes alone. So Lake Superior, for example, you have this really kind of localized projects relative to things like Lake Huron, Erie, and Ontario, where there's much more consistent coverage. You can also have highly variable detection probability. So this can be due to number of receivers, like I said. So here in Lake Champlain, each circle represents a receiver station, and different parts of the lake just have different numbers of receivers. We also have things like zebra mussel coverage on our receivers. That can really impact how well that receiver is going to detect fish that are tagged. And then also just differences based on space and time. So there's a lot of research that's shown things like the thermocline can influence detectability. And I'll point your attention to the map here on the right. So right now these circles represent what we're considering our maximum detection range, about 1 kilometer, but it might be as low as getting down to about 250 meters. So that's a huge change in the detectability of an individual location, and so that can really influence our interpretation of where fish are, if our ability to detect them at a given site and across sites really changes. And I got to really see that firsthand with some of the data from Lake Champlain. So this is data for an individual fish. We'll call it Fish 23. The positions on the y-axis here represent the receiver stations on the lake. They're arranged by latitude, and it's just where did this fish show up over time. Next, I want to bring your attention to these two locations, Inland Sea North and Inland Sea South. So these are these two receiver stations here. And so you can see there's this large gap between these receivers, but we have enough coverage in those locations that we have an idea when fish get into this area, this inland sea area. And with this individual, we can actually see it showed up early on, then got detected down here in the bottom, then showed up again at the top. And there's not a lot of detections in between in that time because there weren't really many receivers there. So by detections alone, it looks like this individual used that part of the lake about 19% of its total time that it was detected. Now, if we look at from the initial point to the last point, that goes up to 47% by duration. So that's a huge difference in the space use for this individual based on how we're looking at these data. And that made me think, can different movement models kind of improve these estimates for regional space use? So thinking about this area as being an entire region when detection data are limited. And I want to emphasize limited here because that's really limited could be a lot of different things, but in the acoustic telemetry standpoint, that's differences in detection probability, which can be driven by a bunch of different things, not just where receivers are, but if you think about things like the mussel coverage and all the different soundscapes around each location that a receiver is positioned at, you can have a lot of differences in the detectability of fish throughout the system. So I tried to address that question by using simulated data to assess the accuracy of multiple different movement models. So we created 100 virtual animal tracks in Lake Champlain using random walk functions. So this is an example of one of these simulated tracks. You can just see how it was developed over time. And then we have the receiver stations placed throughout the lake. And based on where the fish was detected and where the receivers were placed and where the simulated track went, we were then able to generate a simulated detection data frame similar to what you would get from an actual tagged fish. And then we assigned the positions to different regions, so similar to what Dr. Murphy was talking about earlier.